So hello everyone. Today I'm with my friend Oscar, and we're going to talk about genetics. But before we do, I just want to reintroduce a few concepts, give you a few definitions that are important in order to move ahead with the concept of genetics. So let's get to it. So if we remember well, when we're talking about DNA, we know that DNA is genetic material, right? So DNA stands for deoxyribonucleic acid, and that's the matter, that's the substance we find in any cells except the red blood cells, but in any other cell, prokaryotic or eukaryotic. And the difference between prokaryotic cells and eukaryotic cells, we know that eukaryotic cells do have a nucleus, and in that nucleus we have where the DNA is. Now, if you remember the first few lectures, I mentioned that DNA, we saw that DNA is organized in different, there's different levels of organization of DNA, if I can say it like that. So the first thing, of course, as we know, right, is the so-called double helix that's like this. And this double helix is linked between the two strands of the helix, one strand right here and then the other strand right underneath. Those two strands are united together, they're held together by hydrogen bonding, right? So we know what hydrogen bonding is. It has to do with an attraction between certain atoms that are not physically interacting with one another. They're not touching, but they're attracted to one another. Well, the whole genome, the whole part, the whole bit of your DNA inside your cells is held together by hydrogen bonding, so super important. But that is the basic level of that organization. Now, when we look at these double strands here, we know that these double strands are interacting with one another, and they're interacting with one another by their different components of each strand. So each strand has nucleotides, right? Nucleotides, so N nucleotides, and these nucleotides in DNA come in, there's 4 forms of them. There's 4 types of nucleotides. So we have something called adenine, we have cytosine, we have thymine, and finally we have guanine. Well, of course, when we're referring to DNA, we rarely name every single one of those nucleotides. So basically what we do is these nucleotides actually go by their first letters, right? So when you're talking about DNA, we'll often talk about the A, the A's, the C's, the T's, and the G's, right? But you know now that they refer to these nucleotides. Now, the other thing that you need to know, you probably remember, is that adenine always interacts with thymine through 2 hydrogen bonds. There's always 2 between the 2 of them. And cytosine always interacts with guanine and through 3 types of hydrogen bonding. So when we're looking at DNA, of course, we're looking at those here, and those strands here that we have, of course, we would have A, T, G, C, C, G, etc., etc. And those strands are held together via hydrogen bonding, as I just explained. And of course, when we're thinking about DNA, we're thinking about huge amount of information. This is a very, very long molecule. Just think about that. That if you put all the DNA inside one tiny cell, the nucleus of one tiny cell, if we put all the DNA on a table, that would measure about approximately a meter, right? So if you could put all that DNA together, that's held into one tiny nucleus of the cell. This is really amazing. Now, one thing that we know that exists and we need to take into account is the DNA and the sequences, and parts of these sequences, and sequences, when I'm talking about sequences, I'm talking about all those C, G's, T's, and I'm drawing right now a random sequence right? No particular order. And then now I need to only write one sequence because you know that A interacts with T, G interacts with C. Now you know what's on the other side of that sequence, don't you, right? We know that. Now, when we're looking at DNA, we often hear about genes, right? So genes is in DNA, and what are genes exactly? Well, before I get into the gene part of it, I want to explain why the concept of a gene is so important and why it's so central to biology. And here's why. We know that DNA makes us, right? So I have brown eyes, I have a certain height, I have certain attributes, certain genetics to me. I look like my parents, I look like my sisters. You know, there's things that you could tell genetically. There's a lot of things you can't tell either. You know, you don't know if I'm going to be afflicted by such and such disease one day or whatever, right? You don't know, and I don't know many things about myself. But my story, or my potential story, is written in my genes in a way. There's what you can see, and there's what you can see, like my attributes, but also what's inside it we can't see. That's written inside my genes. And there's a name for those two. Okay, so when you're looking at what you see, so say that a flower is blue. Okay, so we know a flower is blue because it has the genes for the blue color. We are referring to something here, and what we can see. So we would say, well, okay, this flower has a gene, or of course genes, and that genes will result in some color, right? In that case, I said blue, right? So genes, color, blue. So the measurement, what we see here, is called a phenotype. Okay, phenotype. And that phenotype is what can be measured, right? So if you look at your eyes, or brown or blue or green or hazel, whatever, this is the phenotype. So if you look in a mirror, or you look at your friend, somebody, you can measure the phenotypes, right? The height, all of these things. So that's a phenotype. Now, what you have inside your cells that say, you know, the color of that flower will be blue, that's written in a code, and that's in the genes. And this is called the genotype. Okay? So the genes themselves, what is written in those genes is the genotype. Now, what you can see now is the genotype, of course, will influence the phenotype. That's not always true, but it is a reality. The genotype, what you have written inside your genes, will and can influence the phenotype. Now, let's see what a gene is, and that's an important part. Okay, so let's erase all of this. Now, let's come back to my sequence here, right? So I have my sequence, and that could be a very, very, very long sequence. And in that sequence of DNA, there's part of that DNA that is useless, if I can say it like that. I don't like to say useless because that's not always true, but let's say that it doesn't serve any purpose for now. Okay, so let's just park this and accept that some parts of DNA might be less important than other ones. Doesn't mean it's really less important, but let's say less important as far as we're concerned for now. And some other parts of this DNA will result in a phenotype, right? An expression of those genes. Okay? So what is a gene? A gene is a portion. It's a portion of DNA. It's a portion of DNA that will lead to a protein, and that protein, or the proteins, will lead to the expression of that information, the genotype, the phenotype, okay? Why am I saying this? Well, here's why. We know, and we will see in this course, a little bit further down the road, that DNA, at some point, leads to the formation of RNA. And that RNA leads to the formation of a protein. or in fact actually proteins. Okay? So a gene at some point will result in a formation of proteins by the cell. These proteins will have a function. That function will lead to the measurement of something, which is the phenotype. Hopefully you're following all of that, okay? So all we need to know now is that DNA is important because it leads to the production of some proteins. And those proteins have a function inside of the cell, okay? So that's an important part right now that we need to keep in mind. Now, let's move a little bit ahead and look at a little bit more complicated things now, because we want to dive into genetics. Okay, so let's move on. So we have our DNA molecule here. It forms this helix. It's, it has a certain structure to it. It's a helical structure. Remember, it's connected via the different bases, right? And when we're talking about DNA in a cell, well, DNA inside the cell is never really organized just like that. It's organized a little bit better than that. There's different degrees of compaction depending on what the cell is doing at any given moment. Now, what we know is that before the cell is just about to divide, right? So that's in mitosis, right? We have, during this compaction cycle here, the cell moves inside the cell cycle, right? So we go from G1, and then we go to S, and then we go to G2, and then we move on to mitosis. And in mitosis, we have all those other phases, right? We have prophase, prometaphase, metaphase, anaphase, and telophase. And we described those in the previous video. So please remember that because that's an important stuff. Now, when we're looking at the S phase of a cell, what happens during the S phase is that the cell, in order to form two daughter cells, it needs to duplicate the DNA. So basically, the amount of DNA in a cell at G1 will become double by the end of the S phase. And in order to separate the cells, we need a mechanism that's going to separate this DNA correctly and perfectly into two daughter cells. So this is why we have all those phases here. Now, what I have here is the organization of DNA into chromosomes. And remember that we see chromosomes only at a certain time of the cell's life, right? It's not always like that. We don't always see the chromosomes at any given moment inside the cell of the life of a cell. We see this only during mitosis and actually only during certain phases of mitosis. So please remember that. And if you're not sure, please go back to those notes and verify that by yourself. Now, what we know is that by the end of all of this cycle here, just when the cell is just about to enter into metaphase, actually in anaphase, where the chromosomes are separated, we will have the chromosomes and we will have the duplicated chromosomes side by side. So we have one chromosome here, one right here, and then we have another one right here, right? And those are called sister chromatids, right? So this is sister chromatid 1, sister chromatid 2, right here, right? It forms the X, and in the middle right here, this section that we see here is called the centrosome. Centromere, not centrosome, centromere. So we have the centromere right in the middle. So this is what unites the two. Now, what you need to know is that for every chromosome, and in human beings we have 23 different chromosomes. Actually, we have 23 pairs of chromosomes, okay? So, let's explore this a little bit more in detail, okay? So we have 23 pairs of chromosomes. So we have chromosome 1, 2, 3, 4, 5, etc., etc. And now remember that the chromosomes are organized in a way that the longest of all those chromosomes we have is called chromosome number 1. And then another one is just a little bit smaller, is chromosome 2, 3, 4. So the chromosomes, as we can see them, they get shorter and shorter and shorter and shorter. So we have 22 pairs of these, plus, of course, we have the 2 sexual chromosomes, they're called the autosomes, either the X or the Y, right? So males, the genotype for males, so what we have, the chromosome that defines like male cells is XY, one chromosome X, one chromosome Y, and then so that's male, and then female we have XX, right? So we have those two here. Now, this is where I want to go to, is not so much to define what male, female is. What I want to say here is that I want to bring a concept, the concept of alleles, because that's important in genetics. So now we know that we have 23 pairs of chromosomes, right? We have 23 pairs. So we have chromosome 1, number 1, and then we have chromosome 1 here, and that's chromosome 1, the other chromosome 1, they come in a pair, right? So this one here was inherited from your mother, and this one here is a paternal chromosome from your father, right? That's why we have a pair. When you were a zygote, which is a cell that was just fertilized, you had a set of DNA from your father in the sperm cell, and that sperm cell entered the egg that had another set of 23 chromosomes, and they paired together and brought us these pairs of chromosomes. So this is true for every single one of all the chromosomes we have. They always come in a pair, 22 plus the sexual chromosomes, which I don't want to get into right now, but I just wanted to say about that. Now, the chromosome so chromosome 1, we say here, if it comes from your father, this one here is a paternal allele. And then here is the maternal allele. So let's say that we have a gene that's right here in the middle of that chromosome. Well, we have the same gene in the other chromosome here. Basically, for every gene we always have 2 copies of it, or at least 2 copies of it, because we have 2 genes, 2 chromosomes, pairs of chromosomes, right? So we have 2 alleles. So now, since we have an allele that comes from your mother, it is possible that this allele is different at some point from the paternal allele. It is possible that in a composition of that DNA, you have variation into the maternal allele and sequences that might be slightly different from the paternal allele. Okay? So basically, the genotype, right? What you have written in that allele, what you see, the DNA part of that, combined with the paternal allele, gives you a genotype. So your genotype is basically influenced by the 2 alleles. Okay? So for example, it is possible for 2 parents that have brown eyes, it is possible for these parents to have blue-eyed kids, right? And you think, well, where is the blue eye coming from? I don't know. Well, you know what? It's because one allele contain the information for the phenotype blue. So the child that has blue eyes has a genotype written, the information written inside the cell allows the expression, the genotype to be expressed in a certain way. Okay? So when you're looking at alleles, the way we define alleles is when you look at pairs, you always have two variation or two copies of the same gene, and sometimes those genes can be different. And of course, that gives us a huge amount of diversity, but that's not the only way diversity is created. We will see in meiosis that diversity can be created, and we say the genetic lottery. You'll see that there is genetic lottery, right? And there's a shuffling of DNA. That's amazing. But anyway, what I wanted to do here is define what the alleles are, okay? And hopefully that's now clear to you.